everyone for holding on to the bitter end here. I'm Morgan Sparks. I'm a master's student at the University of Alaska working on my state fault lab, and I'm going to be talking to you about some of the things we've been doing, looking at thermal-local adaptation, heritable phenotypic plasticity, and embryonic sockeye salmon development. So just to kind of ground us in a little bit of ecology here, I'm going to talk about climate change in Alaska, and I think everyone here is probably fairly familiar with climate change overall, but in Alaska we're really at the front lines of this change. So you just look at this figure here, you can see that last winter in Alaska, the entire state of Alaska was much above average. But in particular, the southwest region right here, that you can see in the bright red, experienced the warmest records on temperature. And that's where my study populations come from. But overall, that's good for us that actually live in the region, but may not be good for the populations that are locally adapted to these extreme cold environments. And in particular, you start to see effects across the landscape, issues with things like fire severity and frequency, and also changes like melting permafrost, and which have effects like changing the hydrology of these systems. And so there are a number of ways that climate change can impact these organisms, and I'm going to kind of couch this in a match or mismatch dynamic. And in particular, I'm going to be talking about phenology a little bit. So we have this potential for organisms being able to shift their phenology and match the environment, which we have good examples from the European tit, but there's also other examples where organisms haven't been able to match their environment, so they're mismatching. And a good example would be this Montana snowshoe hare, where they're actually mismatching their code with the environment that they're in and being more prone to predation. So again, I'm going to focus mostly on phenology today and not talk too much about match and mismatch, and thinking about how climate change might be impacting phenology of the fish that I'm interested in. So those fish are sockeye salmon, and in particular, I'm going to be talking about sockeye salmon in the Bristol Bay region, which you can see in the right-hand map up here. So just a little bit of the general biology of these fish. We get somewhere between 20 and 60 million fish returning to these systems annually. There's 6 major watersheds in Bristol Bay, and you can see Lake Iliamna there kind of in the right-hand side of that map. That's actually the largest lake in Alaska, so that's a pretty substantial sized lake. And what these fish are doing is they're returning to these creeks and streams, and sometimes the lakes themselves. They're spawning in these habitats, and then after the course of incubation and when they emerge from the gravel, they actually out-migrate to these lakes that you can see here. So Iliamna, and then this is the Wood River system, where they actually continue to rear in the fresh water in this mutually shared environment. And after some period of years, 1 to 3 generally, they're going to go out-migrate to the ocean and continue along their life history. You might have heard us talk about kind of in terms of the portfolio effect, where we have this portfolio of diverse habitat usage by many different populations. Also, there's quite a diversity of different history strategies within these populations. And what this helps to do is not only does it buffer against the resource use by a pretty large commercial fishery that's bringing hundreds of millions of dollars a year, taking about 70% of the fish that return, but also against the environmental stochasticity. So this is both an economically important fish, but it's also an ecologically important fish. It's the keystone species in this region, and contributes to organisms as small as caddisflies all the way up to brown bears. So what we were curious with in terms of climate change and kind of these factors of global adaptation and plasticity, we were curious whether or not different incubation thermal regimes had differences in survival and hatch timing. So in particular, were populations locally adapted to their vernal regimes? So if you come from a cool environment, are you going to survive better in a cool environment relative to a warm environment? Then also, were populations, do they develop at different rates? So is there a genetic basis to their developmental rates? So kind of couched within this too, we were curious as to whether or not only do our populations adapt to these different types of thermal regimes, but how does the variation of these thermal regimes play into this? And I'll get into that in a second here when I actually talk about these experimental populations. So what we did is we used 2 populations. They spawn in very different kind of habitat types. So they both come from the Lake Iliamna region. They are just about 10 miles apart from each other across on the landscape. But one spawns and rears on the lake shores of an island in Iliamna Lake, whereas the other spawns in these ponds that you can see here, which are groundwater fed. So the woody island population is kind of warm and variable over the course of incubation, whereas whereas the ponds are much cooler and basically constant over the course of incubation. So when I say that, what I actually mean is this figure that you're seeing here, you can see the woody island population is substantially warmer over this incubation range, whereas the Peter Pond's population is much cooler. And you'll notice too that the variabilities in the thermal regimes are quite a bit different from one another. So if you're thinking about that in terms of plasticity, especially if this one population is coming from a much more variable environment, you might expect it to be more plastic than the other population. So what we did with this experiment is we went out last August, collected gametes in the field. We actually fertilized the gametes and made 10 families for each population. These were actually half-sib families, so they're a 2-to-1 male to female ratio. We brought them back to this airstrip that wasn't very far away from these two sites, and actually flew them about 350 miles from the field to Fairbanks all in the same day. So these fish were fertilized and in-earth treatments in the same day, and then they incubated over the course of 2 to 6.5 months, kind of depending on the thermal regime that they experienced. So this is actually the thermal regimes that we're looking at. We had 5 different treatments. 3 were meant to reflect the Woody Island population, and 2 the Peter Ponds population. And just to kind of briefly summarize these treatments, I'll draw your attention to the MIROC treatment here and the 8-degree treatment. And what these are actually climate change scenarios. And these are based off of downscaled climate model predictions specific to the sites that these populations came from. So this is basically a 2099 prediction for what a thermal regime would look like for that population. And the same being for the 8C population. Now, average and colder just observed temperature regime for Woody Island, and then the 4C is the observed temperature regime for Peter Pond. And this is actually what these treatments look like. And the thing that I kind of wanted to emphasize here is we actually replicated our family at these tray levels. So we actually had 4 trays with a little cup from each family. So we had 4 replicates per treatment. So these are the experienced thermal regimes for 5 different treatments. I'm not going to belabor this too much, but the thing that I really want to emphasize here is that you can see these variable regimes. We actually changed our regimes on a daily time step, so the kind of an average daily temperature. And that's fairly rare for work of this type with sockeye, or with salmonids in particular. And so we measured a bunch of different traits as part of this experiment. I'm mostly going to focus on survival and hatching timing today. And as far as survival, we expected these populations to be locally adapted, so higher survival in your natal environment, lower survival in foreign environments. And then in hatch timing, we expected them to be plastic. We know that temperature is kind of a master variable when it comes to development in these ectothermic organisms, but we did expect them to be genetically different in how they were plastic to one another. So I'm going to jump straight into the results here. And what you'll notice is that we're going to have our regimes, our treatments on the bottom, and they're going to run from warm to cold. And then I'm going to show you on the y-axis the different kind of variables that we measured. And I'm going to also show you kind of these population-specific responses, but also family-specific responses. So the first thing we'll look at is survival. And what you'll notice here is that these 2 populations really survived at pretty different rates. But we're interested in local adaptation, so we're expecting survival to differ between treatments. And if you'll notice, especially these median values, they really didn't change very much from treatment to treatment. If we look at that as a reaction norm, in particular, it becomes even more clear, right? We've got pretty straight treatment-to-treatment differences among populations. Now, you'll probably notice that there's quite a bit of variability in those measures that you saw before. So if you look at this from a family perspective, you'll see that families did fairly similar things as these populations did. That makes sense. But there's a tremendous amount of variability among families. And to kind of emphasize this point, so we've got families, continuing to do these similar things across treatment, so they don't appear to be locally adapted. I will point out, and I'm sure your eye is drawn to this Woody Island response here, and this is indicative of a locally adapted response. But overall, out of 20 families, this is the one response that we had, and we have a number of foreign and natal treatments. So this is definitely not characterizing kind of the overall trend that we observed. So to kind of transition away then from this local adaptation survival perspective, we're going to transition down to phenology, and we're going to look at kind of the phenological responses to these different treatments by these different populations. So if we walk our way through these treatments, I'm going to show you kind of these are our 2 populations here, kind of the differential populations were very, very plastic. So this took about 75 days to hatch in the warmest treatment, took about 189 days to hatch in our coolest treatment. But you also notice if you're looking at these two populations, they didn't really change that much relative to one another in these treatments. And again, if you look at that from a reaction norm, you'll notice that indeed they did basically the exact same thing. So that was kind of surprising for us because I'll remind you, we expected them to do these different things because they live in such different thermal, or they incubate in such different thermal regimes. Now, that being the case, what if we look at this again from a family-specific response, right? So here we are looking at families again, and what you'll notice in particular in this average, these cold regimes, these were our most variable regimes, and you'll notice that there's quite a bit more variability in these families among these regimes. And so we have quite a bit more variability spread out among these families, and if we look at this again as a reaction norm, you'll start to notice that there is some differences in the slopes of these families. So, that being said, our plastic response was about 115-day difference, right? So we have a pretty tremendously large plastic response. But, our kind of G by E response here that we're looking at is about 3 to 10 days difference rate order change in families among these different treatments. So it's far, the response that's being dictated by treatment is a lot larger than these G by E responses. And now, that is definitely a small change, but this is validated by the linear mixed effects models that we did look at these responses through. So we know that these G by E responses are important potentially for adaptation to change. And so this is kind of one of the more important findings coming out of this research. And again, we validate the kind of these G by E responses that we expected to see. So to kind of summarize the results from what I've been talking to you about, we were curious about the genetic and environmental impacts on development. We saw no population level local adaptation in either in hatching or survival. We did see that the populations and families were very plastic, so about 115-day difference between our warmest and coolest treatments. And then I'm going to kind of just emphasize the fact that these populations were very plastic enough to even overcome these 2099 predictions for these specific to these populations, and that these G by E interactions may be important for adaptation in the face of climate change as well. So with that, I just want to thank everyone for listening. Thank the Western Alaska LCC for funding this project, and I'd be happy to take any questions. Thank you.
Sure. So, trying to actually form a measure to someone is making those things split up.
Sorry?
Trying to split up. So,
sure, yeah.
Have you got any reason to perhaps extend it to the other metrics of development?
So, we thought about doing it in this format. I think it would definitely be interesting because emergence ability too is the more kind of ecologically relevant trait. Right. That, especially in that colder population, we're talking about a 250-day rearing kind of period. So, we don't really have the resources in our lab to do that right now. Especially since we just did this in like a room with some troughs, right? We don't do it at a hatchery. It would be super neat to look at if we had those resources. I think that would be one of the other things. Other studies have done that looking at emergence. I will say too, emergence though is also kind of, it's a more kind of difficult to define trait, right? You're either in or out of the tank, but if you're emerging from the gravel, it's much more difficult to say, well, is that fish just coming from luck or is it actually staying in the gravel? So. Anything else?
There is no difference in plasticity among the populations, right?
Sure.
But do you see any differences in the activation of plasticity in the populations?
I don't think, not with what we're looking at here. The interesting thing too about these environments is that, especially the woody island environment, those fish may actually not be adapted